Now let's move on to talking about something called virulence and how we measure that. I mentioned the word pathogen before. A pathogen is some kind of microbe that has the ability to cause disease. So we would say that microbe has pathogenicity, some ability to cause disease. Some microbes cause disease really easily, and some microbes really only cause problems under very specific extreme circumstances. So we would say they are not very pathogenic, or they have low pathogenicity. So another word we could use to describe that is virulence, the degree or the amount of pathogenicity, or how good that microbe is at causing disease. So some microbes would say we'd have very low virulence, and others we might say have very high virulence. So how do we measure that? Can we say how much higher or lower with numbers? Yes. There's two ways we measure that. One is the ID50. ID stands for infectious dose, and 50 stands for 50%. So what this means is the number of cells or viruses or endospores that cause active infection, so causing disease, in 50% of the population. So we're getting kind of an average response of a population, whatever group we're studying. These could be lab animals, these could be people if we're describing the spread of disease. So let's discuss what this number means. So it will be a number, we're talking a number of cells. A very low ID50, say 10 cells to cause active disease in half of a population, that means it's a dangerous microbe. So a low ID50, that's bad. A tiny number of cells causing disease means it's more dangerous. So we would say it has high virulence. So low ID50 means that very few cells can cause disease, which means it's very high virulence. If we look at it the other way, a high ID50 means it takes a ton of cells to cause disease, so that pathogen has low virulence. Let's look at some examples. Here we have a table, some different pathogens, viruses, and bacteria, and their ID50. So what this means is for hepatitis A virus, it takes 10 to 100 viruses to cause active hepatitis A in a host. Something like norovirus, it only takes 1 to 10. So a single virus can cause active infection in an average host. So we would say norovirus has higher virulence and hepatitis A is lower virulence. Let's look at some examples of bacteria. E. coli and Shigella, these are similar, 10 to 100, 10 to 200. So 10 to 100 cells in a host body, or 10 to 100 E. coli cells inside of a host body is enough to cause an active E. coli infection. Now remember, this is a specific strain of E. coli. This is not the kind we're working with in lab. This is 0157, which is an infectious type of E. coli. So we don't have this in lab. So then let's talk about the other one, LD50. This is what we call a lethal dose. The ID50 tells you about how much it takes to cause infection, but not how deadly it is. And it also doesn't work well for things like chemicals or toxins that are measured in concentration and not in numbers of cells you can count. So that's when we use LD50. LD stands for lethal dose. 50 stands for 50% of a population. So the LD50 is the amount of something that it takes to kill half of a population. So that gives you kind of an average response. Let's look at some examples. So these are some different toxins produced by different bacteria. This is botulinum toxin, Shigella toxin, and staphylococcal enterotoxin. These are the concentrations that it takes to cause death in an average host. So botulism, 0.03 nanograms per kilogram is the amount we're looking at here. Shigella toxin, 250, and staphylococcal enterotoxin, 1,350. So which one of these is the most powerful toxin? It would be this first one, botulinum toxin. How can I tell? Because it takes the tiniest amount to cause death. So a very low or tiny LD50, it's bad. That means only a tiny amount is deadly. A very high LD50, that is less bad. It means it takes a lot of that toxin to be deadly.